Tenakoto, and welcome to another biochemistry highlight for the day. Today's highlight is based on an article in the New York Times by Jonathan Coram and Carl Zimmer. Let me share the screen for you. They take you through all the proteins encoded by the coronavirus genome. They explain each in very simple language, and I had a good time guessing what they meant in the language of biochemistry. For instance, if we scroll down here, untagging and cutting, NSP3. This certainly sounded like a deubiquitinating enzyme to me. And NSP5, protein scissors, certainly the protease. I was particularly taken by the spike protein, protein S, because this is the site of a simple insertion mutation, 4 extra amino acids, that makes it far better at binding the receptor on human cell membranes and probably accounts for being so very infectious. So I was intrigued to follow that up a little bit more, and they give us the whole sequence, the RNA sequence of each one of these genes. So all I had to do was select that, copy it, go to the XPASI Translate tool, paste it in, the proper sequence, translate it, and I get the whole protein sequence out and can go and find that little insertion there. But if I want to figure out what it does, I really need a 3-dimensional structure. Fortunately, the Protein Databank has a coronavirus feature, so it's convenient for us to find the structures that have been solved for these. Let's go there. So there's the current Protein Databank front page. We can click on the coronavirus's resources. Give it a moment to load. We get handsome paintings by David Goodsell showing us the coronavirus, but we also have recently solved structures, including the spike protein. And that'll show us all the options. We can choose one that we think will be informative. And if we look at the pictures, we've got two options here. One looks like it's the full protein, all 1,273 amino acids, but we also have these up here, which show us just the interaction of one domain of the receptor of the spike protein with the ACE2 receptor on human cells. So if we wanted to see the full protein and what that mutation was doing, it turns out there's a recent publication in Cell from about 3 or 4 weeks ago showing us the structure of the entire thing. They talk about what that mutation does, and that's certainly worth following up with these authors. But they don't actually have a structure of the spike protein bound to the receptor. That is in the this structure up here. 6-LZG. And so if we go to Pymol to take a look at that, let's reshare here. That shows us a little bit of the spike protein on the right and the ACE2 receptor enzyme on the left. So if I'm interested in that interface, how do I go about visualizing that? Well, one way is to build up a selection which focuses on just the interface. And you can do that by starting with a selection which is just the ACE2, this one here. And let's duplicate that so we can play with it. I get a new one. Now let's modify that and ask for everything close to it, but not itself. So use the around one. Residues within, let's say, 4 angstroms. And so that adds some off of the spike protein that are close to that one. Let's modify again to get just the ones on the spike protein. Modify, restrict to a selection, and I've already selected the, oh, no, I haven't. So I need a selection which is just the spike protein. So let's rename this to something convenient. And let's make a new selection, which is just the spike protein. There we go. And modify those interacting ones now to restrict just to the selection, which is that chain. So that gets us ones just along the selection. I can now work back the other way, get a selection just on that side. And I can add the two together to get the entire interface. If I then show side chains for that as sticks, I get all the interacting side chains. You can zoom in on it, get rid of the annoying pink dots. And within that selection, under actions, ask it to find the polar contacts for me and get a group like that. Use the color buttons to color them something you can see and begin to inspect. We've got a crucial tyrosine residue here, kinds of dives into a pocket. If we show a surface on that, we can see the complementarity of how the spike protein fits into the receptor and begin to guess where we might build drugs to combat that. So it's a, you can use Pymol to explore any interaction. And if you were thinking about what to do for drug design against coronavirus, this might be a place that you would start. Till next time, be well.